Well, this is transmembers versus intracellular receptors. And then now we're going to look at how they usually alter gene expression. For example, sex hormones are steroids, which pass through cell membranes. I'm just going to read every slide.
Just read it.
Read it.
You're doing great.
There you go.
So, Miss Gussel passed her HL bio exam because she's really smart. Where were we? We're talking about transmembrane versus intracellular receptors because it's so fun. That's why. Okay. I'm going to make this as simple as I can without changing you. But the idea here is if it's transmembrane, okay, it's in the membrane. And it passes all the way through the membrane, since transmembrane, so that it can initiate a response inside the cell. Okay, cool. But there are other receptors that are, inside the cell, hence intracellular receptors. And in both cases, the ligand binds to the receptor and then initiates a response. But let's talk about some differences real quick. The intracellular variety, this little guy, it's inside the cell. Therefore, the ligand has to move into the cell before it can bind to the receptor. So you guys need to know. You should already know. Man, that traffic's awesome. You should already know what kinds of molecules can pass through the phospholipid bilayer, but I'll tell you anyway because I love you. Yeah, hydrophobic molecules. So the sex hormones are steroids, and as steroids, they are hydrophobic and therefore can easily pass through the membrane where they can bind to the receptor. Now, these guys will then move through the nuclear pore where they grab onto the DNA and change gene expression. So, estradiol and testosterone are going to alter gene expression, and that is what brings on all the sex-specific characteristics, thanks to the fact that those hormones are hydrophobic. Okay, check it out, get your notes, and then move on. Now, transmembrane receptors need to be stuck into the membrane, duh. But how do they do that? Well, they need to have a hydrophobic portion that likes the hydrophobic tails of the membrane. They also must have a hydrophilic portion that likes the cytosol and the extracellular environment. So what does it mean to have a hydrophobic and hydrophilic region? Well, simple. That means the amino acids in this part of the chain are hydrophilic, and the amino acids in this part of the chain are hydrophobic, and therefore it folds and tucks neatly into the membrane. And again, the difference is that the receptor binding portion that binds to the ligand is out here, and that causes a little conformational change that initiates an intracellular response down yonder. Okay, cool. Oh god, here it comes. This is the hard stuff, but don't panic. It's okay. Right here in the middle here, the big point is that, you get your ligand binding to the receptor, and then it's not like poof response. Well, it is very fast. Don't get me wrong. Cells are so efficient, but there's a series of changes here, yeah. And that series of changes is called a pathway. Like, for example, the Krebs cycle is a pathway. It's a series of changes. Now, this is a signal transduction pathway. So your ligand binds to the receptor. We get a signal transduction pathway, which all adds up to a response from the cell. And we have to do examples, and this is when it gets nasty. Okay, signal transduction pathway examples. First up, the OG, the G protein coupled receptors. All right. G protein coupled receptors, the OG, they basically are receptors that have a very close association with this very fancy protein called a G protein. And it is a G, it's true. And what the G protein does is it has this sort of complex series of subunits, the alpha, the beta, and the gamma. And so to keep it simple, the beta and the gamma subunits can detach. And they're the ones that go off and carry out the cellular process, and so when we talk about the signal transduction pathway here, the way it works is the ligand binds to the G-coupled receptor. And then the G protein disassembles, and then the subunits initiate a cell response. So that's our pathway. In between, I skipped over sort of an important series of events here. Is this stuff GDP and GTP? You can think of this like ATP and ADP, except instead of an A, you have a G. And I'm not being silly here. This is guanine instead of adenine. And then this is triphosphate and diphosphate. And very much like ATP, it can be built up and broken apart. Here's your phosphate coming off, just like ATP. And so it has a very close association with this G protein and helps to drive the signal transduction pathway. Okay, more. We're going to do two examples here. We're going to talk about how blood sugar can be increased and decreased. Now, let's talk about epinephrine briefly. It kind of, I know this is all here. You're going to have to tune it out for just a beat. But epinephrine, as you know, is our fight or flight hormone. When epinephrine is released, the body kicks into hyperdrive because you need to either run away from something scary or fight something scary. In either case, you need energy to fight or fly, right? And so you're going to need some sugar. Yeah. So what does your body do? It takes all of your sugar stores. You got glycogen stored, in this case, glycogen stored in the liver. And that glycogen needs to be broken down into glucose so you have energy to fight or flight. Yeah. So how does it work? Epinephrine binds to a receptor. And it's a G protein coupled receptor. And it's in the liver cell. Epinephrine binds to that receptor. Now, the unique part here is that after our G protein does its business, it signals conversion of ATP, not used for energy here. This is unique. ATP is going to get a little bit of a modification, and this ATP gets modified to cyclic AMP, which you can think of as sort of like a catalyst that then interacts with this enzyme. Sort of like think of it as like activating this enzyme. And what does this enzyme do? It breaks down our glycogen to release glucose. So, transduction pathway. After the binding of our ligand, epinephrine, we get G protein activity, which leads to ATP formation, no, ATP transformation into CAMP, which then gives us activated enzymes, which break glycogen down, releasing glucose so we can kick